With regards to the agenda this morning, again, I'm going to give a brief introduction and some, updates as to what's going on. I was hoping that Angel would come online and give us an update as to the status of the in vivo experiment. And then we're going to get an update from Alex, Zhongfu, and Alison on some of the assays that they're doing now. And then Sue is going to give us a chemistry update. Then the last time we had some concerns about the formulation that was used in the in vivo efficacy studies, and so we had gone back and done some formulation work looking at HCl salts and mesylate salts, and that looks quite promising. So Ahmed is going to give an update on some of that result today. And then one new addition to the agenda, we added Julia, so that she can give us an update on some of her plans for her 3D screening for some of the compound in the DIPG line. And at the end, there's an opportunity to discuss any burning issues. Hello, was somebody about to say something? Oops. Okay. So what I thought I'll do, last month, just to give the group an update, we're kind of in the final phase of, addressing some of the actionables for the CTIP grant. And so I thought I'll just do a quick summary of some of the actionables that were left the last time, and we're actually approaching them. So just to remind people, one of the key actionables was to demonstrate, compound-dependent modulation of some PD marker. So for that experiment, we decided to look at this hepcidin model, which is currently ongoing at Charles River right now. So as soon as that becomes available, we'll make that information available to the group. But we're thinking that this hepcidin might be a good PD marker for, addressing whether when compound gets on board, are you actually affecting, readouts for ALK2 target engagement. One of the other goals that we had actually committed to at the beginning of the program was to look at ex vivo, identify biomarker from the brain, but as we thought through the project, we realized some of those may not be trivial, particularly in a clinical setting, getting biopsy. So, that was an ambitious task at the end. We thought it might be worth abandoning that goal. So, the next thing was to do the efficacy study. And so some of that work is going on in Angel and Chris's lab. So I hope that somebody just called in. Hope that's Angel.
No, it's Ben.
Okay. So what's nice is that we had started that efficacy experiment, and so I'm hoping Angel will get a chance to give us an update as to where that experiment stands. But then some of the other goals, like assessing tox before the efficacy study, we had done that. The compounds are well tolerated, so that was completed. We have the backup series, which is the Blueprint, and we are also considering the AMI series, which solves the hERG issues as also a backup. So we had actually made more than enough compound. We had completed that goal, and we got enough crystal structures for the program. So I think we had addressed, kudos to the team. We had addressed most of the goal for the program over the 2-year life of the granting part of the project. So I just want to give the team a big kudos for that. Thanks to everybody's effort for getting a lot of that done. And so we managed to get the CTIP report in on time. We're supposed to give a presentation to the CTIP committee on November 19th. And I think I had mentioned to the team the last time, Alex had suggested that we ask for an extension. So that's one of the things we have requested from the committee, that we get an extension for the grant so that we can finish up some, a few experiments. And so with regards to updates to the team, I just want to remind the team again that for all of the 5 lead candidates that we have, the scale-up are still ongoing. It's all on track. I mentioned to you the last time that 2 of the compounds are going to be delayed because of one of the starting material that's required for the synthesis. But the delivery of 2117 and 2163 should be on track for the end of the year. Another actionable we had last month was to complete the data set for 2163, one of the compounds that's looking really active in some of the DIPG lines. we hadn't had a complete data set for that compound, so we now got the rat PK then. Okay, and I'll give you an update on that. And I think currently they are running the mouse dose escalation study. So that would now give us the full complete data package for all 5 lead compounds that we can now start using to kind of see which one rises above in terms of some of the other studies we have planned. Again, as I mentioned, the DIPG efficacy study is ongoing in Angel's lab, and also Diana Martins and Chris Jones' lab was supposed to be doing the Belgium model. We haven't heard from Diana, but it would be nice to get an update as to where that is. With regards to DIPG lines and understanding some of the mechanisms, I know Alex's team has been doing some of those studies, and now it looks like Julia from Charles River is going to also be looking at some studies in some of these 2D and 3D models. So Julia is going to give us an update on some of the planning for that later in the meeting. Also, we are pursuing some PROTACS strategies just to help us better understand, based on knockdown, which of these lines are truly dependent on ALK2 for their growth. Another effort that we have ongoing is, again, the backup series effort from the Charles River team, along with some of the design of the PROTAC. We'll get an update from Sue on some of that. That's looking promising. And again, as I mentioned to you, Charles River is currently performing the hepcidin model, and we're doing a pilot study with 2009 right now, so we'll let the team know what's going on there. Like as I also mentioned, so we have formally requested a no-cost extension from the CTIP, so we'll see what the feedback is, but I don't foresee any problem, you know, as long as it's no cost. It's okay, as long as we're not asking them for money.
So normally, how long are you, is there a time specific, how long are you asking?
Till March 2020, yeah. We asked for no cost extension until March. Okay, with regard to the PK update, happy to report to the team that the PK for 2163 rat looks really good. I mean, the bioavailability is 74%, and you know, 10 mg per kg dosing, we're getting nice exposure. The profile looks very good. And I just have the mouse data here just for comparison so that people can know. So I was quite happy to see that there was no, you know, surprises here in terms of, you know, bad PK all of a sudden for one of our exciting leads. And then what I did on the next slide is now, for the sake of comparison, I put all of the 5 leads here so people can see the relative profiles. The 3 compounds on the top, those are the most CNS penetrant, and they all have very good oral bioavailability in mouse. They have very good oral bioavailability in rat. So that bodes well for the next phase of the studies in wanting to do tox and stuff. And then the 2 compounds which are moderately brain penetrant to the bottom, they still have decent bioavailability. So I think we're in a good place in terms of the rodent profile for these types of compounds going forward. Again, with that list of compounds I just showed you for PK, I thought, you know, it's nice to show people what these structures, just to remind people what the structures of these compounds are, some of the features that they exhibit, and then the profile for these compounds overall look really good, I thought. Okay, so with regards to now timeline, again, I mentioned we asked for no cost extension. The actual grant for this phase of the project was supposed to finish at the end of this month. Okay, but like I said, we asked for no cost extension. We've achieved most of the goals I think we wanted to accomplish for the grant. Now at that time, we have now 5 lead candidates, and subsequently we have some of these other more costly studies that we have to do, dog PK, rat dose escalation for tox and tolerability, and more important, some dog telemetry to really de-risk some of the profiles, some of the exciting compounds. So I think we're in a very good place. And then we still have cancer to the bottom, but we still have the backup program, and I think the backup team is now starting to make some nice inroads in terms of properties of some of these compounds. So you'll hear some of that from Sue. All right, so that's basically the intro. If no one has any questions, is Angel online? Angel, are you online? Angel? Okay, so it looks like Angel is not online, so the data has to be really good. So why don't we go on then to Jean-Fu and Alison and Alex update? I'll just pull those slides.
Who's presenting?
Okay.
All right.
Can you email Alex? Can you email him just to see if he's
Okay.
please?
Yep.
I don't know. I look, but I don't know.
I will do. Yeah, it's posted.
Yes, Alison sent me the slides. I'm just looking for. I just uploaded.
They weren't presented.
Oh, I actually uploaded them. I didn't.
They're still in your email.
No, I actually downloaded it. I just need to. Oops. This one.
I can't find their slides.
I have it open.
Yeah, just read open. Open it.
Huh?
Just go ahead and open it. Just open it, read open. Read it.
Yeah.
There you go.
So you guys can see that? Anything? Oops. So you guys can see that? How about that?
Yeah, there you
Okay.
All right. Yeah.
All right. So you want me to drive? I'll drive.
go. Yeah, that's great. So just an update on the routine screening for the cell-based assays. So if you can go to the first slide, please. So 5 compounds have come through for the routine nanobreath screening in the last month. So just a couple of those have shown reasonable potency. So the M4K 2D74 and the 2163, which I think is the second batch. And the 315 is a borderline sort of potency there at 113 nanomolar. So I've taken those through to the ALK2 dual luciferase, and none of those are really showing any potency in the off-target ALK5. So if you can move to the next slide.
I was just going to say before you move on, that's interesting. The 3105 and 3110 are the 2 compounds from the second blueprint patent. So it's interesting that they are a bit weaker than the first.
No.
They're less weight, bidentate hinge, because they've got the aza-indole at the hinge. They clearly are. They were weaker in the primary.
Okay.
Because the initial compound, they were extremely potent as well as
Yeah.
Next slide, please. So this was just to check PROTAC activity. The 3 PROTACs we have, particularly the M4K3102 has got a really nice potency in the ALK nanobreath. So took those through to ALK5 also, and that's showing that there's really no off-target activity in those compounds. Next slide. This compound goes through to the luciferase assay in the C2C12, so conformational ALK2 binding in those assays, and we really see that all the low potency compounds, they replicate in the C2C12, so it's nice to see that there's confirmation of those compounds. The 2163, I'll have to repeat that again because I'm listed on the video, so we should have data for that in the next meeting.
You're breaking up quite a bit, Alison. I don't know why that is, but
Okay, can you hear me clearly now?
yeah, that's a little better. It's still a little muffled, but much better than a few minutes ago.
Yeah.
Which one is
Okay, so the ALK2 dual luciferase.
It may help if everyone else mutes. Go.
Okay.
It looks like it's better.
Okay. Okay. So the 5 lead compounds for the M4K series have taken them from nanobreath through to the C2C12 dual luciferase assay, and we see that the binding potency confirmed there in the CTC12 ALK2. And just the 2163 compound, which needs repeating in the next run of the C2C12 ALK2 assay, but unfortunately there was a misdispense. Okay, next slide. And this was something that we flagged in the last meeting. So we're seeing that the potent activity that we see in the wild type is said to be Blueprint-scaffold compounds. Sorry, there's quite a lot of on the line.
Yeah, Wow. Wow. Yeah, it's really bad.
Someone didn't mute here.
Everybody mutes, and then that should help.
Okay, so
We can still hear somebody. Still.
Tourism like it. You better find out who it is.
okay, that's better. Ed. That's much clearer now. So can everybody still hear?
Yes. So Arish and Max, could you guys mute?
You guys can control it though.
Yeah. Oh, we can. Oh, we have total control. Wow, that's good.
Sorry about that.
Okay, sounds good.
Okay, great. Okay, so just checking the binding of the blueprint scaffold compounds. So this was something we flagged in one of the earlier meetings that we had. So, sorry, we've lost the slides.
On their side.
Yeah,
it's their side. They muted everybody.
Okay, go ahead now.
So you've muted us. We're only temporarily unmuted.
No, we can hear you.
Muted himself. We can hear you. Keep talking.
Okay. So we noticed that the binding of the M4K 3078 and the 3095 wasn't showing the same potency. So for the whole series, the binding potency is the same for the wild type and the mutants Q207, 328B, and 206H. But the G356D for the 3078 and the 3095 shows no activity. So I'm just going to hand over to Ellie, who's done some structural modelling to try to explain that interaction. So Ellie, are you?
Yep.
So the 3001 is actually quite interesting because it binds all the mutants, which is what we want, but not the wild type.
Okay.
But we should check. That's worth double checking.
Yeah.
Which one's that?
The second column.
Okay. 3100.
That's more from the other series, though. The 3100 is more from the other series.
Yeah.
Peridot. Yeah, so that's not surprising. It's hitting all the mutants.
But it's more active against the mutants than the wild type. That's interesting.
Other people reported that, but I never really believed it. So this is borderline.
I wouldn't say it's massive.
No, it's not
We need to
massive,
look at
no,
those
but
confidence intervals to
Yeah, it's
see.
more active against the G356D and the G328B.
That's
Yeah.
also one that's quite active at ALK5 as well, though, isn't
It
it?
is.
Okay.
Yeah.
No,
It's
that's
not
true.
that
Yeah, yeah.
selective. This one, we're a little bit.
Do you want to come in?
I'll just go from here if that's okay. Next slide. Okay, so this is just a quick model, and it's based off the structure from 2007, which has a similar kind of back of the pocket binder. But in the G356D mutation, obviously the aspartic acid there is going to stick out far more than the glycine. And the effect of this is probably going to displace the D354, which is normally flipped to the right, but in this particular mutant, it's more likely to be flipped to the left, as shown in this figure. And that, of course, is then going to bring us to within less than 2 angstroms of the sort of triangular group there on the, sorry, I'm not a chemist. Profile group at the back of the pocket. And that's going to make that compound far less potent in this mutant. But obviously, in all the other mutants, glycine is present, so the aspartic acid is flipped the other way, and so there's plenty of room for it. But that kind of explains why the 3095 and the 3078 aren't going to work.
That's really important for chemistry, though, I guess. And how important is that group for binding, for potency? Can we do without it?
Yeah.
So the other thing that we've noticed before is that there's a water binding network at the back of that pocket.
Yeah.
And so that group there isn't really contributing to that much because it can't form any hydrogen bonds. So the trimethoxys that you often see at the back there are often taking part in that water network, and you get lots of water
It's the
bridging.
carbonyl. Tell me that is.
So the carbonyl there is, because it's close enough to your K235, that's about 3 angstroms. So that is important, but then that's also further away from your aspartic acid. It's just the propyl group there that isn't.
So have we made just the methyl?
We haven't, no, because
should do that.
in the blueprint pattern, cyclopropyl was the most common one, so that's why we stuck with that. But there's also that in what we think is the BLU, whatever the number is, it has a tetrahydrofuran.
Right.
So it's got an oxygen in the ring.
Yeah, that's interesting.
So it
That
may be
probably
worth
wouldn't fit.
So anything that might
no, it might,
contribute to the water network there or make bonds might.
yeah, so it might be worth making one.
Yeah, that's too small, I guess. This data's telling us, otherwise it wouldn't bind to a nutrient.
No.
Certainly lots of our structures are good enough to show the
they've
water,
got,
so you
they've
can have a
got
look.
the same, they've got it as a carbonate.
Okay.
So there's an oxygen, then a group. But they're quite a nightmare to work with because they're quite unstable. Okay.
Okay, that's everything that we have.
Okay, thank you. Any other questions from Alison and Alex team?
Some chemists once told me they don't like ketones.
Okay. Oh, just look at the faces, oh my God. And they still do.
What was that? We missed that. Was that a question?
Move on.
Okay. All right, thanks, Alison. Up next is Ahmed. We're going to give you an update on some of the formulation work that we have done in light of some of the issues we had seen with the formulation and the efficacy study. So let me come upload your presentation.
Just hold on.
Yeah. Get to the full screen. Yep.
Okay, so I'm going to just update you on the M4K 209 formulation work. So we normally use the DMSO formulation. I'll show you the composition of the formulation in a minute, but let me just tell you. So that formulation we use for our R&D work. So it is very good in that sense that it dissolves, almost 90% of our compound at the dose level we want to dissolve. So this is why we use this R&D formulation. Normally, we haven't seen any problem with this formulation. However, this is the second time we are having problem with the orthotopic brain models. So, like we had some issue in Spain when they were dosing this formulation, the mice in vehicle were not doing so well. And we had similar experience when we were working with Calgary team. Also, we had some issue with brain orthotopic models. So then we try to change the formulation to a different formulation. We know methylcellulose is quite benign formulation. It works very well with mice. So we want to use methylcellulose. But the problem is methylcellulose is not going to dissolve the compound. So the compound is going to be, is going to be a suspension. So there will be dose-to-dose variations and stuff like that. So what David and Meth they did. This has the basic amine group, obviously this compound, and they did salt, 2 salts. One is the hydrochloric acid, the other one is the nuclear salt. So we checked the solubility of those salts, and they are easily dissolvable at about up to 100 milligrams per kilogram. dose formulation. So, so this is the study design. Having said that, so so the two salts were made. We dosed them at 25 milligram and at 100 milligram per kilogram PO, and using methyl cellulose formulation. Historically, so this is the formulation we normally use. Historically, we dosed it in 5% DMSO, 47.5% PEG, and then 280 water. And that is the R&D formulation we use for most of our studies. Okay, and then we collect plasma from 3 mice at each time point. So the time points are 30 minutes, 1 hour, 2 hour, 6 hour, and 24 hour, and then do a LCMS analysis. Okay, so and here is the data. So forget the green bar for now. This is the historical data. What I'm focusing on these bluish and grayish colors right now. Just focus on those 3. Those are the free base in methyl cellulose, hydrochloric acid in methyl cellulose, and the mesylate salt in methyl cellulose. Basically, I did an AUC analysis also this morning. They are almost identical, right? So the exposure for all those 3 formulations is almost identical. However, if you compare it with the DMSO formulation, which this is the green bar right now, the AUC is about 2 times higher, which is not unusual. This is not the first time we are seeing it. Usually, this DMSO very soluble formulation usually gives you very high exposure, and that's what we have seen, and this is not a surprise for us. So yes, it's about 2 times exposure. So this is the data. So I think we can go ahead with the methyl cellulose formulation and do the next experiment. So the next slide here, if I can go, sorry. Okay, so next slide is the 100 milligram per kilogram. Again, as expected, we see quite decent exposure. However, we don't have a historical data here with the DMSO PEG formulation at 100 milligram per kilogram. So I couldn't compare. So maybe we can do a bridging study just to see how it looks like. So we don't have the 100 milligram per kilogram. Or the other thing we can do, or we should probably do, look at the brain exposure. We did not do a brain exposure with methyl cellulose formulation yet. So this is something I think we should be doing just to close all the open angles or open sites. So then we'll be all kosher, I guess. So yeah, so this is the picture says 1,000 words, I guess. So this is the formulation. You can see these are the 2 salt, HCl salt and the mesylate salt. This is the free base in the same dose level. This is 100 milligram per kilogram solution. Actually, the 25 milligram per kilogram was very similar. It was also a suspension dose. So yeah, that's how it was dosed. However, as I said before, they are giving similar exposure even at the cloudy solution was this one here, and then the other solutions were giving slightly higher exposure at the initial time, but overall AUC didn't change significantly. Okay, so that's pretty much all I have to say. This is the number. You can see only one thing I'm going to point out in the number. So when you are comparing the 25 and 100, it is slightly super linear. It's not exactly linear, meaning if it is linear, it will be exactly 4 times higher. Even at the AUC level, it's not exactly 4 times. It's slightly higher, which is not. It is close, but not unusual also. So that's all I have to say. And this is the conclusions. Thank you.
Yeah, so this is good. So I mean, and you could see we end up picking the 100 mg per kg because that was one of the doses we did in the efficacy study. And so the fact that it's a nice solubilizing concentration, Angel did mention that methyl cellulose is a vehicle that he knew is most tolerated. So this bodes well for any future efficacy study that we want to do and avoid that DMSO formulation, which looks like in some tumor models it's compromising. I mean, we have done that formulation in other tumor models and it's fine. But then we've also seen in some tumor models, some orthotopic models, that formulation can actually be a problem in some of the vehicle groups. So it's good to be flexible. It's nice to see that the salts are extremely soluble, so that shouldn't be an issue for dosing. So we're looking forward to any efficacy studies. I mean, we've gone ahead and made, you know, a little larger batch of one of these salts. We've actually targeted the HCl salt. It was interesting whether you should talk about that little delay, whether the subtle delay.
Yeah, so there is a, so if you look at the, so mesylate salt, I didn't go into the details here. Usually mesylate salt, when you look at the 24-hour time point, it is giving us a exposure even at the at 25 milligram per kilogram, which is slightly higher than the other forms. But the hydrochloric acid formulation was giving us initial boost. So, you know, like if the drug is Cmax driven, maybe hydrochloric acid salt will be favorable. But if you think about long exposure, mesylate salt is giving you a little bit more exposure at the later time point. So that is the thing. But one thing also from our experience, we know when you're giving a high dose of mesylate, it's not usually very good for the mice. It's usually repeat dosing for longer time. It can cause also a gastric lining problem. So that's why one can avoid. If you can avoid mesylate, that's also good. So I would recommend right now to go ahead with the HCl salt, unless we have to change into mesylate salt.
Okay.
Question. This is Nicole from Charles River. The other thing that can affect this is crystallinity of the compounds, and that can affect solubility. So do you know when you're comparing all of these salts whether they're crystalline or not?
So
all of them are actually very, very fine powders. Yeah. They're uniform in that way. There's no particular distinction from one salt form to another, and the free base looks actually like the two salt forms, so very comparable.
We should probably to XRPD or techniques such as that can tell you whether it's crystalline or, because ultimately as you move forward, you're going to need to go with crystalline material, and then
we have to do those types of
but if you think about it, if it is already soluble, if it is already dissolved, then it not really it doesn't have a huge impact, right? If it is not soluble, then we could discuss like what would be the right form. But at the dose level we tested, at least they are all soluble. So I don't think it's going to make a huge difference. If it is not soluble, I'm just repeating myself. If it is not soluble, then we can go back and analyze all the form. But it looks like at least at 100 milligram per kilogram, it is soluble.
I guess eventually, I mean, whatever, you know, salt form we pick going forward, if that's the case, I mean, we got to do the proper crystalline workup. So we'll get studies done, crystallinity and properties. But right now, we just took the amorphous compound, free base, we transform it to a salt. Looks like it might also be amorphous. I mean, so, but we need to probably follow up with nice, you know, crystallography studies of some of these salt forms.
So Methan,
okay.
We have a pharmaceutics group here at HCRL who we can get to, we can at least talk to them about salt forms, and we've also got formulation people who can advise on going forward as you go further down the line into the clinic. You may well need to look at different salt forms and different formulations.
Yes, I mean, we just pick, typically at this stage sometimes we just pick two salts, and we always pick HCl and mesylate to kind of do pilot studies. But I agree, eventually we'll probably need to do a full salt form workup, get a company to just screen all these different salt forms and see which one of them actually might be the best. Okay, but good point.
Yeah, no, I think you and I will chat about this, the plan for the next 4 months, because it may be something, it's probably something that we want to start addressing a little bit further, especially after our conversations about the future path.
Yes. You know, I mean, yeah, because this is also good when we start doing tox, you know, tolerability studies in tox and rodents and stuff. The formulation becomes very important too. in terms of what sort of achievable concentrations with dose escalation. So all those are going to be critical things to consider in the next phase. Okay. Thank you. Any other questions for Ahmed? Any other comments? Any other suggestions? If not, who's next up? Julia? Okay. So we get to Julia's slide. Oops. Julia.
Hello?
Yeah. Oh. Hi, Angel.
Hello, how are you doing? Sorry, I joined late because I didn't know the time today. Sorry.
Okay. No problem. That means you have really good data for us, I guess. Okay. Yeah. Thanks for joining. We're going to do Julia's presentation and then we'll come to yours, if that's okay.
Excellent, thank you.
No problem. Okay. So hi, Julia, you up? Hello? Julia?
She may be on mute.
No,
Uh-oh.
I don't think she's there.
So is Julia online, you know? Add a mute. Hello? Sue?
Sue muted too.
It looks like it. We'll ask him. Oh, you haven't done Sue's presentation. Hello? Hello? Huh? Okay. Angela, are you still there? You lost everybody?
Some reason we lost everybody. Doesn't sound like anybody's on.
There's a chat. Someone is
I'm
saying-
here. I'm here. I was silenced.
Maybe we should go with that.
Maybe.
Okay, let me pull up Angel's presentation. I probably should share.
I can mute it.
So Angel,
Yeah, I have to share.
so how do I do this?
Can you hear us?
Hello?
Can you hear us in Charles River?
Yeah, you guys disappeared for a few minutes. Yeah, but we can hear you now.
Julie's not in the room with us, but
Okay, and it doesn't look like she's online, so we're actually going to go to Angel's presentation, and then we'll come to Sue after that. So we're going to share. So we just stop sharing, am I?
Yeah, I need to share.
Angle, yeah.
Stop share and then mute here.
Okay, Angel, you should have control now.
I'm sharing now. Am I sharing?
Yeah.
Okay, there you go. We have, let me, can you see this?
Yeah.
So I'm gonna update the results. Actually, the study is finished. We have used the DIPG 7 model. We know this is going to be 100% take rate with a median survival between 70 and 100 days. The tumor is going to be diffuse and infiltrative with these two mutations, 3.3 and ACVR1. Later, Balaguer did all the study. So the study started in July. We inoculated how many mice? Like 48 mice, and the treatment started like 1 month later. And there were 4 groups: the control group, the 100 milligram per kilo group treated for 4 weeks, 50 milligram per kilo treated for 4 weeks, and 100 milligram per kilo. We wanted to treat for 8 weeks, but finally we did it for 6 weeks, okay? So we wanted to do two objectives. First, to see what would be the evaluation at the end of the treatment. So we sacrificed 3 mice from groups 1 to 3 at the end of treatment. That means at 4 weeks after treatment. And then remaining mice were used for the survival study, and the endpoint finally was 25%, not 20, as says here, weight loss due to disease. So these mice, the remaining mice, were left untreated after the end of treatment until endpoint. group 4 received not 4 more weeks, 2 more weeks. So total 6 weeks treatment here. The end of treatment samples were samples for PK, which I will send to you. We will ship them to you. And this is brain and plasma. And we also process some samples for to analyze tumor burden. To analyze tumor burden, what we do is we measure the amount of ACVR1 mutant DNA by digital PCR. And this is the result. We don't see significant differences between the treated groups and the control group. This is preliminary, but we will repeat this again next week or in 2 weeks, and we'll try to confirm. These are 3 animals, these are 3 animals, these are 3 animals. So this is the amount of tumor measured as nanogram of mutant DNA in each of the brains of these groups. This is the follow-up of mice, the weight of the mice. So as you can see here, the red ones are the controls. So the red ones, we started treating them with vehicle here this day, exactly this day, because we didn't start initially by mistake. So we started here by day 43, and they started losing weight because the vehicle is toxic. They lost weight. We finished all treatments here, day 67, 68, I guess. And we didn't give more vehicle here, right, later? We didn't give more vehicle to the control mice, and they kept on losing weight, as you can see here, because this is the disease. They are sick, they lose weight. And the mice that are treated, they lose weight, but not that fast. So they are sick, but they remain a little bit better than the treated mice. This is the mean weight loss in each group. And as you can see, the 3 treated groups, they overlap, and the control groups go below the other groups. And this is the survival. All the control mice died with a median survival of 82 days. And the survivals for the other mice is like 1 week, between 7 and 10 days long, or 7 and 9 days longer than the control group. And the P is significant compared to control group. We will apply here the von Ferroni correction. I think it will be significant enough even after the correction of this von Ferroni. So we are, in one side, we are satisfied by this study. We are replicating what we obtained with the LDN drugs. On the other side, I don't know, we are not satisfied with the vehicle. We think that was difficult to interpret sometimes.
I don't know if you missed, so we had a little discussion just before you came on about the vehicle. We actually found a nicer vehicle system that's much more solubilizing. It's methylcellulose in water. The compounds are very soluble. So, I mean, if we have to ever repeat any efficacy study, that might be the vehicle to go with. I mean, you're getting very, very good exposures with it. So, yeah, so that's a presentation you missed just before you came online. So when we post all of these slides, you should probably get a chance. I'll send them to you anyway. I'll send the slides to you with the vehicle study that we have done. Yeah, but we have a much better vehicle, we think, for the efficacy study that hopefully wouldn't compromise the animals.
I also have the immunohistochemistry for the staining. Let me see where it is. It is here.
Yeah.
Here. We have these slides showing the infiltration of the tumor in the brainstem of controls. Oh, this is 50 milligram per kilo. So these animals were treated with the 50 milligram per kilo dose, and this is the infiltration of the tumor at the end of treatment, which is a lot. I mean, the mouse is full of tumor, the brain is full of tumor. All these brownish things are the human nuclei. This is the same for the 100 milligram per kilo. So the animals are with the tumor there. Where's the control later? Here. The controls are like the same, you see. So there is no difference in tumor infiltration. And we try to quantify this a little bit, and there is like a similar number of human cells. So I don't know where the difference in survival comes from, but definitely I would like to maybe repeat the study with the vehicle.
From SGC Oxford.
Again?
Hello, somebody.
One of the slides that you show, you try to quantify the mutant ACVR1 in terms of like the DNA content, like as like hand in hand with your IHC, right? We'll be able to try to basically distinguish, for example, quantify the amount of human cells in just like in terms of like the DNA, for example.
Sorry, I didn't understand very well because it was not very good.
Any loading any common protein like beta-lactin or like FDH or anything, because I think those probably would be more robust compared to like trying to, I mean, specifically quantify the mutant ACVR1.
Yeah, we did that. You mean the whether the quantification of DNA is better than the immunohistochemistry? This is the quantification of mutant DNA. This is digital PCR to do that. And we have these results. We don't see difference, clear difference. Although this is all preliminary, we need to confirm, review the data better, and let you know what is the final result. But this is what we have so far. So this is the amount of mutant ACVR1 DNA in the controls. And you don't see a lot of difference between the treated and the controls. Even this one could be, I don't know, a layer. We will repeat this sample too next week.
So previously in some of the other studies, you do see stark differences between?
We do. We've seen differences with one study we did recently with Chris Jones, and with another study we did with anti-STAT3 drug, and we didn't see differences with the LDMs in the communications biology paper. There is no difference between the groups, even though there is a slight difference in survival. We didn't. I don't remember very well if we did this quantification of mutant DNA in this LDN, but I guess we did. Maybe we didn't use it for the paper, but we didn't see a difference. I don't know. Is Diana there? Diana or Chris are there online? Maybe she can confirm. She's not. Okay.
Yeah.
So, any other question?
I think so. You said a comment about the vehicle had a problem. So do you think the if everything is same and if you repeat the experiment with a different vehicle, do you think the outcome would be better or different? Do you think?
I don't think so.
Okay.
I don't think so, but I really want to not having this toxicity in the control mice, because then it is more difficult to interpret the data of the survival, because you have to like balance the days you treat the mice with the days in which the symptoms will start. So the symptoms are also weight loss. So if you have a toxic vehicle or a toxic drug, it's difficult to interpret, especially if you want to keep the treatment for a long time. So that's why I would like to do, and I know very well carboxymethylcellulose is like the perfect vehicle because there's nothing,
Yeah, so the compounds are very soluble in carboxymethylcellulose as dissolved, so that would be ideal. I think the other key piece of, something I know, I'm still a little bit concerned that we give the animals a break on the weekend. And so maybe if we get to repeat, this was a good pilot run, but if you have to repeat this study, just to give it closure, it'd be nice to actually be on the target all the time for the life of the experiment. So the question is whether or not when we're given breathing time on the weekend, it just bounces back. And so, that's something else to think about in the next experiment.
But anyway, Medvin, the drugs were toxic, so it's not possible to keep the treatments for 4 weeks if you don't stop like a couple of days, because the weight, they lost weight very sharply, and with these 2 days they recovered a little bit, and we could keep the treatments longer. So I think
but do you think it's vehicle related? That's the question. I mean, so one of the concerns we have is the vehicle related. So if we eliminate that vehicle and we don't see any weight loss, then we probably might have a cleaner experiment to really make a call. I mean, I don't know. That's what we're thinking, but unless you think differently.
So are we doing PK/PD in this experiment?
Yes, we're going to get samples,
It's just PK.
Yeah.
But no PD.
Well, PD is the histo and DNA here.
Oh, that's okay.
Yeah.
That's the.
The models, these are the kind of PDs you gotta get accustomed to.
This is not the inhibition of phosphorylation or actual
No.
mechanistic
No.
PD.
No. Something we could do is I'm sure if somebody wanted a phosphosmad or one of those kind of experiments. Okay, thanks, Angel. This is, a lot to think about. Any other questions for Angel? Any other suggestions? Comments?
No, I was just thinking, if you had a phospho-SMAD readout or something, it would be nice to show that you're actually engaging the target while not having these effects. I mean, that's a definitive outcome.
So with these three samples, I will send it to you. There is like frozen tissue. There is frozen tissue, and also paraffin and plasma. So you can check the markers for the PD.
Yeah, I think that would be.
You can also check on the PK at this after 4 weeks.
Yes.
Yeah. I think that would be very useful to help us interpret this.
Yeah, I think so.
Awesome. So we'll do that. All right. Thank you.
All right. Thank you.
Thanks, Angel.
Thanks.
Oh. So next, Sue, you're up.
Pardon me?
We need to get back control.
View options.
So
Do I need to do
Angel,
it?
you have to give us.
Do I need
You have
to
to
do
give
anything like a stop sharing or something?
Yes, you have to stop sharing.
Oh,
Yeah.
sorry.
Thank you.
Sorry, I don't know how to do it now. Where is this?
Before we go into the details of this, there's more doing angles on the head of a pin.
Is it to this up? Yeah.
Oh, here. The red one. Okay, I'm done.
Yeah.
Okay, thanks.
Pull us back up. Thank you.
Thank you.
See it. Switch one in the middle. See it. Okay, there you go. Sue. Yeah, wait, this. Sue. Right. Go to this later. Yeah. So that one over the second one, yeah. Yeah. Okay. Sue? So you're up?
Yeah, but the slides.
Okay.
10 minutes per same talks.
What do you think?
I don't know, I'm not sharing yet, wait.
Yeah, it is. It goes. Is that, you're fine?
No,
Yeah?
I can share. I should be able to see it, right?
Yeah, that's it.
Okay, sorry.
Oh, you're logged in too?
Yeah.
Can you see the slides?
Yeah, no.
Yeah. Okay.
No,
It's okay.
We have to.
I can only see the screen that says join audience, share and invite others.
We don't have the share screen option, so wait.
Give us a sec. Wait. Somebody gonna click on that share thing. Yeah, is that it? Where's that blue share thing that is the PMP bottom? That's a sharing option of that one. Yeah, there used to be a blue share thing there, click on over here. Yeah, is it this one?
Should be able to do it now. Yep.
Okay, so now we go. Okay, how is that? Are we good now?
Should be.
Can you see now?
Lovely, thank you.
Can you see?
Yeah, now, thank
Okay. Awesome. Okay.
Well, next slide then, please. Okay, so just to, as we discuss each month, we're continuing to prepare analogues within the 2 blueprint patents. We've made a number of compounds over the last period. We've got some further compounds coming along. So the other thing that we've been doing, and we discussed briefly, we're working with an AI company called Atomwise, and they've often helped us to try and identify compounds within the 2009 series that actually have improved selectivity over ALK1 and ALK6. So they've done some initial screening and that's virtual screening, and we've started making the compounds. Also done some further work on PROTACS, and we've been looking at a strategy to improve the cell permeability and potentially get brain penetration. Within the biology area, and I've not got any slides on this, but we did discuss it with guys this morning. We've been looking at some biology to help with the PROTAC work that's been carried out at the SGC. As you've already mentioned, there's work ongoing in Shrewsbury within Charles River on the hepcidin model, and hopefully the results will be reported in due course. And also Julia's been doing some work, but obviously she seems to not be online, so apologies for that. So go to the next slide, please. So this is just the sort of introduction, again, the usual introduction to the Blueprint series. As we know, the lead compound from this series has only moderate permeability and efflux consistent with its poor brain penetration. And we're trying to address the potential issues of high molecular weight and high pKa, which could well be contributors to this poor CNS penetration. So our current plans are aimed at overall improving the general physchem properties while maintaining the potency and selectivity. Next slide, please. So probably the most promising compound that we've prepared recently is this compound M4K 3093, which has good ALK2 potency, retains good selectivity over ALK4 and ALK5. And looking at the physchem properties, we've compared to 3007, we've reduced both the molecular weight and the pKa while retaining acceptable log P and TPSA. So we've been remaking a sample of that, so we've completed that resynthesis, and some compound was sent to Methvin recently, and they've looked at permeability, and it retains good permeability. It has low efflux ratio and moderate microsomal stability. So that's going forward into PK to see whether in fact it does improve the brain to plasma ratio. So hopefully we will see an improvement here. Next slide, please. So we've made a few additional compounds. So the right-hand end is actually the piperazine rather than the ethoxypiperidine present in 3007 to maintain that lower molecular weight. So we're looking to try and find alternatives to the piperazine there. And we've looked at, looking at these piperidines and aminopyrrolidines. So all three compounds have been made, but we've only got data on the first of those. Unfortunately, that wasn't active, but the other two compounds should be tested in the next run. Next slide, please. Within the second blueprint patent, we've made a number of compounds. We've made two of the three compounds that we said. Synthesis is ongoing of the third one, which is compound 68, shown on at the bottom there. The first two compounds do have moderate activity. And I saw earlier that there's also some data from the nanobret assay, which shows that they're not as active as the compounds from the first blueprint patent. So, however, these compounds are obviously much smaller, so they do have potential improvements from the molecular weight perspective. And we're looking to see how we might be able to learn from these series and incorporate something into some of the other series as well. So next slide, please. Yeah, so these are just a few of the things that we're looking at. So that fluoro compound M4K 3093 at the top there, we're looking to see what other groups we might be able to incorporate into that position. We've looked at some docking, and it looks like slightly large groups, because we've got a relatively small gatekeeper, slightly large groups may well be tolerated. And then we're also looking to see, we've got compounds like 3095, which was quite an active compound, which had a slightly modified core, and see whether we can actually cut that compound back further with bidentate hinge interaction, whether we can actually get reasonable activity with that. And at the bottom, again, we're looking to modify again the tail group. So again, can we use the smaller groups from the second blueprint patent, but also take out the terminal basic center from the piperazine on the right-hand side and reduce the pKa there. And it's just a few examples of some of the sorts of compounds that we're looking to make. I think that's probably my last. Oh no, it's a bit on PROTACS. Next slide, please. So there's a little bit on PROTACS here. So the first couple of compounds actually showed quite good activity, and we've seen that 3102, which was first of the PROTACS, had quite good activity in the nanobret assay. So we've got a few further compounds that we've prepared here. These have all shown around 100 nanomolar activity, so slightly less active than RBC. This is RBC data, yeah. So we're working on some plans to try and reduce the number of H1 donors within the linker to try and improve the physichem properties to help enhance permeability. And that work is currently ongoing. I think that is my last slide.
Thank you. Any questions for Sue? Okay. If not, I think, sorry? Did someone have a question?
Are there thoughts on delivery of PROTACS? That'd be the convection treatment that David suggested.
Well, ideally, we're trying to look to see whether we can orally active in brain penetrant.
This morning was
and the Arbanest data.
Yeah, okay, sure.
Yeah.
But yeah, that's always my concern when people come forward with these sorts of approaches.
I guess we started this for a different reason, which is to use them as tools to knock down the protein and look at the biology, but there is the potential to make them brain penetrant and make them into future medicines, but that field is evolving still, I guess, and we can be part of that and we can follow that. And we talked about that this morning, it could be quite exciting, but we just need to think about that, how it balances with the other work that we're doing.
Yeah.
I had a quick question, Methvin. If the compounds that we're making with atom-wise, we talked a while ago about getting them screened for ALK6 so that we could look at this selectivity hypothesis. Is that going to be possible with RBC?
Yeah, I mean, Owen, you have enough cash left to screening? Just kidding. Yeah, we should be able to screen them at RBC.
Okay, we can talk about that offline, but we should also check that was still possible. Thank you.
I think it is.
Yeah. Yeah, okay. Thank you. If not, thanks everyone for the updates. As I say, Julia was unable to join, but she sent a plan for what she's planning to do for this GD experiment. I'm just going to post them at the WebEx site so people can go and have a look. It's just 3 slides. Okay, and so hopefully she'll give us a full update at the next meeting. All right, and I'll keep you posted with regards to the decision for the extension. I don't think we'll have a problem with the extension that we want for the project, but when that's formalized, I'll let the entire team know. All right, thanks again, and we'll be in touch. Thank you.
Thank you.
Thank you.
Awesome.
It's interesting that your link is all exactly the same.
You just mean right at the solvent edge. So once you're out, and it doesn't really matter what angle.
Yeah.